Hi, my name is Avi Balsam, and I'm from Highland Park, New Jersey.
Hi, I'm Jonas from Switzerland.
Hi, I'm Sarah from Mexico.
Hi, I'm Victoria de Leon from Mexico City. In this video, we're going to present an innovative approach based on synthetic data generation for the modeled kinetics of bioluminescence imaging, also known as BLI. In a few words, BLI is an increasingly popular method which has been used in modern biological research applications such as gene activation tracking, tumor genesis, cancer treatment, disease progression, and so on. That has been, of course, very helpful for the advancement of modern healthcare, but this experimentation comes with a cost. This cost is animal testing, as several laboratory animals, generally mice, have to be genetically modified, constantly injected, or both, to achieve ideal experimental results. Having the knowledge of this, we decided to tackle the problem efficiently through a synthetic data generation program made in Blender, a 3D computer graphics software, to simulate reality through different visual representations based on bioluminescence kinetic curves, which basically model what occurs in the test subjects, mice, during the induced chemical reaction. With this approach, it is not necessary anymore to experiment thoroughly on animals for picturing certain behaviors. Also, for giving much more potential to our synthetic data, we developed a Python module capable of producing various noise models that mimic image sensor behavior at various lighting conditions, representing common problems that inevitably happen during imaging, and opening great possibilities for using this data for training an ANN, or artificial neural network, capable of obtaining the real light signal, which of course, once done, can be applied for a wide number of imaging scenarios.
Now let's talk a little bit about what bioluminescence is and how it relates to imaging. Bioluminescence is a type of chemiluminescence where a living organism has the capacity to emit light. This happens as a result of a chemical reaction between luciferins, that are bioluminescent substrates, and luciferases, which are enzymes that catalyze the oxidation of the luciferins, producing luminous energy. BLI is the technique used for detecting and revealing the signal of the visible photons released at specific wavelengths, and as a result of this chemical process. Through a series of scans, every phase of this reaction can be modeled with a kinetic curve based on time signal intensity that shows the progression after luciferin injection, the peak point of the light emission, and then the decay. They are extremely helpful to determine the best point of after substrate injection to image these subjects, as well as to analyze the efficiency of different luciferins in order to determine the best choice. It is very important to model them because each behavior changes depending on the unique characteristics of the living organism that is tested, which is one of the main reasons for the experimentation in multiple laboratory animals. Now, we will present the methods we used for developing our solution.
To generate our synthetic data, we used Blender, a free, open-source software for 3D modeling. We chose Blender because it is reasonably easy to use and understand. We hope that our discoveries will open up the world of synthetic data generation to those without the resources to run some of the more computationally expensive methods that have been developed by other researchers. The system we have developed may be useful to scientists inside and outside the biophotonics community who do not have time to scale the steep learning curve Monte Carlo simulations present. Additionally, our method is extremely computationally inexpensive, making it useful for generating large amounts of synthetic data for machine learning algorithm training or multi-frame data for kinetic analysis. In Python, using Blender's scripting library to automate the process of data generation. In brief, the program turns this mouse into this class, generates a light-emitting blob, turns it into code, and randomly places and scales the blob inside the mouse. After setting up the scene, the program uses an approximation of the luciferin kinetic function to set the light emitted by the blob for each frame, and approximates the mouse's natural bioluminescence with exponential decay. Simple, right? We used a robust object-oriented structure to allow for changes and modifications to our program. The end user can modify the number of blobs, the minimum and maximum intensities of the blobs, the scale of the mouse, the light emitted by the mouse, the position of the blobs, and a host of other parameters. Additionally, the user can replace the mouse model or the blob we provided with another model of their own choosing, or add more objects into the mix. We see this program as a foundation for further refinement and research of computationally inexpensive data generation methods. The program takes only a minute to run and generates a reasonable approximation of real-world data. The fact that this method is so inexpensive means that we can efficiently generate a wealth of training data for machine learning algorithms and other data-intensive processes.
In practice, images taken with bioimaging methods are unfortunately quite noisy. The main cause of this is the camera itself. As the signal, which we need to detect, is quite weak, those machines are very sensitive. But because of this technical necessity, they also capture the thermal radiation caused by the camera. Also, there is some cosmic radiation which causes some almost completely white spots in the images. To simulate this behavior in our synthetic data, we combined two already existing noise types. As one goal of the study was to decrease the usage of animals in clinical trials, we decided to show the whole process on everyone's favorite mouse, which is probably invincible anyway. The raw data, which we produced with the Blender simulations, consists of single frames. So we also need to merge them into a single sequence to visualize the kinetic curve produced by the decaying luciferin. So the script we developed has two parts. First, we noise the single frames. This has two components. We apply Poisson noise, also known as shot noise. This type of noise is commonly used to simulate photon counting in optical devices. As Poisson noise is signal dependent, we can't simply increase the variance to make it stronger. To achieve this, we rescaled the images. On top of this, we added Gaussian noise. This type of noise occurs often in digital images when the environment has, for example, a high temperature. Thus, it is perfect for our needs. The variety of strength is much easier to achieve with this kind of noise, as we can simply change mean and standard deviation, and add, instead of apply, the noise to the images. The last step of the process is the merging of the frames. After that, the data is ready for usage in machine learning applications. To address the specific problem of overfitting, the noise is randomized for each frame.